Hello everyone, this is me Farah Fatma. Today we are going to discuss about the theories that are involved in enzyme substrate complex formation. Theories involved in enzyme substrate complex formation. Theories involved in enzyme substrate complex formation. As we all know that a number of biochemical reactions are continuously taking place in our body, and the substances that are involved in these reactions, they are regarded as substrates. This is an enzyme, this is a substrate. This substrate binds to enzyme, form enzyme substrate complex, then enzyme substrate complex, then enzyme product complex, and finally product is obtained, and this enzyme, this is available for reuse. This is an enzymatic reaction. This enzyme substrate complex formation, how this enzyme substrate complex formation occurs, a number of theories are involved here. Basically, we'll discuss about three theories. Number one is lock and key model. Lock and key model, also called Fischer model. Number two is induced fit model. Induced fit model, or we say it Koshland model. And number three is substrate strain model. In this video, we'll discuss only about lock and key model. Lock and key model. This theory, enzyme catalysis reaction, the theory explains that the active site of an enzyme, this is the active site. The theory says that this active site of enzyme is a rigid and pre-shaped structure. Rigid and pre-shaped structure. And theory says that when this substrate comes in contact with this enzyme, then enzyme substrate complex formation occurs, according to this theory. And this was given by a scientist named Emil Fischer. He was a German biochemist. He gave this concept for the very first time. The basic point of this theory was that the active site is rigid and pre-shaped structure. And the theory also says that this substrate binds to the active site in a very similar way, like a key. Like a key fits to keyhole of a lock. Like a key fits to keyhole of a lock. Here we can say that substrate fits to active site of an enzyme. The mechanism is very similar to that of a lock and key mechanism. This was the theory proposed by Fischer. But the drawback of the system is, the drawback of this theory is that it ignores, this theory ignores about the flexible nature of, flexibility of enzyme. This was ignored, and hence all the enzymatic reactions that are because of the flexibility of enzyme, these reactions, they were not discussed by this lock and key model.